In this video, we will discuss about CRISPR-Cas13 function against the foreign RNA and some of its application. So let us quickly revise what does actually CRISPR, how this CRISPR system function. So whenever this invader injects its genome, and when it's this whenever this invader injects its genome for the first time, the host immune system does is it will take the some part of this genome and integrate it into its own genome system, and this is known as adaptation. So and later on, when this invader invades the genome for the second time, this expression of this genome part that is already been integrated into its system will get expressed, and this will integrate it with the Cas13 system, and then it will this time it will interact this time this complex that is Cas13 system with the expression of this genome interact with this ssRNA that is injected by this invader and helps in uh, breaking down this ssRNA, and this process is known as interference. And the Cas13 system function mainly comes when this during this adaptation process, where this uh, invader genome is get integrated into the host genome. So there are two different kinds of integration occurs. First, when this invader directly inject this RNAs that is in the this part, and whenever the invader injects its genome in the form of DNA, that is in part A. So let us understand this part A, which is most commonly uh, used whenever people use this CRISPR-Cas system. So what happens is this invader's genome part get get detected by the immune system of the host, and then it will be and then using this Cas2 and Cas1 it will be integrated into this host DNA, and in this arrow that can be shown here, and the integrated part is shown on the bottom uh, DNA derived spacer, and the bottom this CRISPR edited DNA, this green section. So this is how this DNA part is integrated. However, when this RNA part when this RNA genome is this injected by this invader, what happens is that right in this case the RT is also plays a role. So this RT Cas1 and Cas2 will form a complex, and this will helps RT means reverse transcriptase. It helps in conversion of this RNA into the DNA, and this cleavage occurs with the help of this Cas2 and RT Cas1, and after this reverse transcription, simultaneously this reverse transcription of this uh, integrated this foreign injected RNA genome will also take place, and after that this reverse transcription of this foreign injected RNA, or in other terms we can call it as complementary DNA, will be injected into this uh, host genome that is shown by this pink or red color. So this is how this CRISPR-Cas13 system works. Now let us understand how this CRISPR-Cas13 system, this is known as CRISPR-Cas13 system, and how this. Let us just quickly go through some of its application of CRISPR-Cas13 system. It can be used in the case of this microfluidic device in order to detect whether a particular RNA is present or not. And so how it will work is whenever this our RNA of our interest is present, it will be then integrated into this. It will be then translated by this Cas9, Cas1, this RT Cas1 and Cas2 system, and simultaneously since if our RNA will be fused with some fluorescence reporter. In this case, example is shown about the GFP, and during this translation we will get this GFP translation will also occur, and we will be able to see the fluorescence signals. So it can be used to detect the presence of RNA using the microfluidic device. The next thing that it can do is it can help us to track the RNA in the cytoplasm. So this Cas13 system can be integrated into the cytosol of the cells, and that will help us in tracking specific kind of RNA or a translation the transcription, whether the transcription is occurs uh, of a gene changes or not, that can be observed using this RNA tracking, cytoplasmic RNA tracking, or in common terms we can call it as epigenetic tracking of particular RNAs can be tracked using this case. And this thing can also be used for this nuclear RNA tracking. In this case, we need to inject one nuclear localization signal that can result in targeting those. It can result in uh, transferring this complex into the nucleus of the cells. The third part is that it can help us to prevent us against some conserved viral RNA sequence. So the conserved RNA sequence will be integrated into the host cell or using this Cas13 CRISPR array, and after that this Cas13 CRISPR array will be translated along with this information of the conserved virus RNA. So whenever the virus RNA will inject, this Cas13 CRISPR system will interfere or break this in viral ssRNA that is injected through is injected as the cause of this infection of ssRNA. So in this ways it can be used as antiviral applications. The last thing that it can be used is for the treatment of genetic diseases. Some of the genetic diseases, there is misfolded proteins that are continuously formed, and these misfolded proteins can be targeted by designing this Cas13 system against those against the sequence of this misfolded proteins, so that this Cas13 system with in forming a complex with this CRISPR RNA, it will target the misfolded proteins that is causing this genetic disease. So in this way it can also helps to relieve some stress on these genetic diseases. So these are some of the applications of this CRISPR-Cas13 system that target the foreign RNAs. If you need to understand more about this CRISPR-Cas13 system against the foreign RNA, you can go to the reference that has been provided in the description. That's all for this video. Thank you for your time.